In this lesson, we'll continue our discussion of the cellular level of organization by looking at a second membrane transport mechanism known as osmosis. In the previous lesson, we had done diffusion. Now, osmosis is like a special type of diffusion to me. What it is the diffusion, give a big highlight right here, of water, right? This is going to be the movement of water. Remember that regular diffusion was the movement of solutes. This is the movement of water across a membrane that is selectively permeable to water. Right? Osmosis takes place across a selectively permeable membrane that is freely permeable to water, but not to solutes. It's only going to let water pass through. Okay, so this is a kind of like a type of diffusion, but it's not allowing solutes to move across the membrane. It's going to allow water to move. As a general rule, guys, we're going to see that the water molecules are going to move across the membrane towards the solution with more solutes, right? So whatever side of the membrane has more dissolved substances, it's almost like a pulling force. The water is going to be pulled towards those solutes. This is going to become important in your subsequent studies in AMP2, not so much in AMP1 when you study how fluids move in and out of capillaries, how urine is made. Urine is going to move through kidney tubules. This concept of osmosis will become important then. Okay, so this is an important slide. Osmosis is the movement of water across a membrane that is permeable to water, right? Freely permeable to water. It will allow water to pass through, but not to solutes. Let's take a look. If we were in the lab, we could actually do an experiment where we have a special type of test tube. So let's start with this first one over here. I'm going to go to just the bigger picture so that we can see it a little bit better. On the side A of the test tube, we have in it water molecules. So those are the small blue dots. Then on this side of the tube, there are some dissolved substances. Let's just say these would be salts. Okay, just as an example, say this is salt. Then the dotted line in the middle represents the selectively permeable membrane. Now, these big salt solutes cannot pass through that membrane, only water, right? This membrane is selectively permeable to water. Let's take a look at the letter B. So, same thing, we have water over here. But notice, guys, we also have much more solutes over here, more salt, let's say. All right, you can see there's more bigger dots on the right side, the B side, than there is on the A side. Once we let the experiment happen, osmosis will occur, which is the movement of water. So from the previous slide, we had said that the water will move to the side where there are more solutes. So in this case, where is there more solutes is on the B side. Watch this. So notice what will happen over time. The water will move across. And now, where is there more water? Look how the volume went up on the right side, the B side. Just to check. Remember the original level was here? Okay, now take a look. They actually put a dotted line for you. You can see the water volume went down on the A side and then it went up. And that's because the osmosis occurred. The movement of water was to an area where there is high solute concentration. So again, if this was dissolved salt, if this was sugar in the bloodstream, this would be a protein. We can call it albumin. There would be more albumin on one side than the other side. The water would move into the blood vessel with the more albumin. So we can definitely see that the volume of the water increased. Okay, we'll talk about this in a moment. Let me just go back to the notes just to make sure we have everything. All right, this is just everything on the same slide. I just broke it up for you before. Okay, all right, so the really important definition again is the diffusion of water, not solutes. And again, the water is going to move to where there is more dissolved solutes. Okay. Again, we're going to go back to this slide in a moment. I just want to go to this next slide here to explain it. So the movement of the water into, say, the B side of that tube actually produced pressure. This pressure we're going to call osmotic pressure. This is the force with which pure water moves into a solution as a result of the solute concentration, right? So it's just the force that the water is moving that we call in this pressure. So if I go back now, watch this. As the water right here. As the water is moving over, it's increasing a pressure. That movement of water is actually like a force. That is known as osmotic pressure. Now, in the lab, we could do this. We could actually push down here. You see they kind of put this metal thing here, and we kind of push down on the water and force it backwards through to the other side. That pressure we're going to call hydrostatic pressure. This is the pressure needed to block osmosis. These two things we'll talk about more again in AMP2, osmotic pressure and hydrostatic pressure. But just think of osmotic pressure as more of a pulling force where the water is pulled to the side where there is more solute, and then hydrostatic pressure we tend to think of as a pushing force where we're pushing the water back. Okay, and again, we'll leave it at that for this semester. As you go into AMP2, you'll look at that in more detail. Okay, osmosis occurs more rapidly than solute diffusion. This is because water can cross a membrane through abundant water channels, meaning we have a lot of special channels that are water channels. We call these aqua porins. Think of aqua as water, and as these are proteins that have a pore in it, like an opening. So we tend to have more aquaporins, excuse me, than regular solute channels. So the movement of water can be very, very rapid. Okay. A couple other definitions. Osmolarity, right? That sometimes they call this the osmotic concentration. This is the total solute concentration in a solution. Basically, what we're looking at is, let's start over here, how much solutes are in solution, right? So we would call that osmolarity or the osmotic concentration. Now, there's another term, and I'm going to apply this in a moment, something known as tonicity. This describes the effect of osmotic solutions, right? Solutions that have different concentrations of solutes. What are their effects on the cells? And we're going to go over three examples of that, where we're going to put a cell in three different solutions that have different osmotic concentrations, right? Different amounts of solutes. So let's take a look as an example. There's something known as an isotonic solution. Basically here, iso means the same. Um, Let me show you an example. Let's say we have a solution, guys, right? So let's just say out here is some water, the small blue dots of water that are in the solution, and then we have some solute molecules. So again, let's just say this is salt, right? These big pink dots are salt. Notice the amount of salt on the outside of the cell is the same as the amount of salt on the inside of the cell. So since the concentration of the solutes is equal, there's going to be no pull of water one way or the other, either out of the cell or into the cell. Very simply, as some water moves into the cell, an equal amount will move out, right? As some water moves into the cell, an equal amount will move out. So this happens to be a special cell known as a red blood cell. It's not like a cheek cell or anything like that. It's a red blood cell. And how do I know that other than it's red? See how it's kind of concave right here? Here's a real red blood cell. Our red blood cells have a little bit of a dip into it. They look like a frisbee, and then they have a little bit of a dip. So when we put a red blood cell with an equal amount of salt in the water, let's say inside the cell, and we put it in a solution that's the same salt concentration, the water into and out of the cell is equal, and the isotonic solution will cause no change in the shape of the cell. It will stay exactly like a frisbee with a little bit of a dip on it on the top here, and there's also a dip on the bottom, believe it or not. So an isotonic solution does not cause osmotic flow, right? It's not going to pull water into the cell or out of the water. A cell in an isotonic solution stays the same size and shape. All right, it's not going to be affected at all. But watch this. The second type of solution is known as a hypotonic solution. So hypo means less than, or here they put below. If we put that same red blood cell in a solution that has less solute, let's say less salt, watch what happens. So we take that same red blood cell, guys, right? We drop it into the water, but notice inside the cell we have solutes, right? One, two, three, say four salt molecules. But notice out here, there is no salt. So what's the rule? The water is going to move to an area where there is more dissolved substances. So since there's more dissolved substances in the cell, the water is going to move in the cell, and look what happens. The cell changes its shape. Instead of being a frisbee with the sag in the middle, it became like a ping pong ball. Right? This is all filled with water. If enough water moves into that cell, this cell here, the red blood cell, could actually rupture. And we have a term for that. We call that hemolysis, right? Hemo means blood, and lysis means to break, right? To break apart. So a hypotonic solution, the solution is a lower solute concentration, right? There's less salt. And as a result, the cell will gain water, right? The water is going to move into the cell because inside the cell there is more solute, more salt, let's say. And again, if enough water goes in, we call that a condition where the cell actually bursts, hemolysis, right? Hemo, blood, lysis, to break down. But we pronounce it hemolysis. Okay? Let's do the opposite. If we took a cell, same red blood cell, let's say, right, and we put it into a hypertonic solution. Hyper means increased or above, meaning that the water is more salty than the cell, right? The water is going to have, let's say, more salt than the cell. Let's take a look. So notice, guys, out here in the water we have a lot of salt, let's say, more than what's inside the cell. Right? Inside the cell we have our same four salts, so that's kind of constant. But look at this. This is like salt water, really concentrated salt water. So what's the rule? Osmosis is going to be the movement of water to wherever there is more salt or more solutes. Since there's more salt outside the cell, look what happens, guys. The water is going to move out, and the cell, as a result of having less water on the inside, is going to like shrivel up. And you can see here, this is a real red blood cell in a salt water. See how you get these little spikes on it? This is known as crenation. If I go back a moment, yeah, right here. So a hypertonic solution is a solution that has a higher solute concentration, right? More salt than what's in the cell. And again, the water then will move to where there is more salt, and that causes the cell to lose water and shrink. And again, we call that crenation. Okay, so that's the end of this lesson as far as osmosis. Again, the important things to remember is that osmosis is the movement of water. And again, the water is going to move to where there is more solute concentration, right? More solute concentration. And again, if we put that red blood cell in an isotonic, right? Iso means same, meaning the salt concentration is the same as the cell and the water. There's going to be no net movement of water. It's just going to kind of be an equal exchange back and forth. A hypotonic solution, right? A solution that has less salt in it or less solutes. Put that red blood cell in there. The red blood cell is going to take in the water and it's going to swell. Potentially it can burst. And then finally putting that red blood cell in a hypertonic solution, right? Much more salt in the solution than is in the cell. The water is going to move out of the cell to where there is more salt. All right, and that is our osmosis. In the next lesson, we'll actually take a look at a special type of transport known as carrier mediated transport.